Hello students, we'll start with second unit. In second units you have to learn on the cofactors and coenzymes, and the second you have to learn about Michaelis-Menten equation and enzyme inhibition. Okay, this video I'll try to give a brief explanation on cofactors and coenzymes. As I told you in the previous video, cofactors and coenzymes, they are part of the holoenzyme. Cofactors, they are non-protein partners. Usually they are inorganic metals, maybe iron, copper, zinc, magnesium or calcium ions. They bind to enzyme and change its shape. The change in the shape is essential for the activity of the enzyme, normal function of the enzyme. Then the coenzymes. These coenzymes are organic compounds. They derive, maybe they are from the vitamins, okay, and also the non-vitamin parts. The organic cofactors derived from water-soluble vitamins like niacin and riboflavin, that is vitamin B2. They have the role in transfer of electrons between the enzyme. That is also essential for the normal function of the enzyme. That's how these cofactors and coenzyme, so they are part of the holoenzyme. So I shown you in the form of a flow chart. Cofactors may be inorganic ions and coenzymes. Inorganic ions, I have listed some of the metallic ions here: magnesium, selenium, iron, copper, manganese, molybdenum and nickel. And in coenzymes, it may be vitamins and non-vitamins, but both are organic that is organic compounds. Organic compounds, vitamins may be fat soluble or water soluble vitamins, and non-vitamin parts, it is ATP, coenzyme Q and glutathione. They may be act as a organic compounds or act as a coenzyme in the functioning of the enzyme. In elaborative, the cofactor is a non-protein chemical compound. See here, I have shown you the cofactor region where it is the schematic representation of a cofactor that binds tightly to the enzyme. It aids in the normal function of the enzyme. This is a holoenzyme. It binds to the inactive form of an enzyme known as apoenzyme. The apoenzyme, that is, when the cofactor binds, it becomes active. And this binding of a cofactor to the apoenzyme make it ready for the reaction, or it is active. Therefore, cofactors are called helper molecules. So these cofactors, they are called as helper molecules. The active form of enzymes, the active form of the whole, after binding cofactor, then the coenzyme. So enzyme is active. So it is a complete, that the holoenzyme. The cofactors can be either metals or, as I told you in the previous slide, there may be either metals or coenzymes. Coenzymes are organic part. Inorganic substance like metal which firmly bind to the enzyme and are unable to remove without denaturing. We call them as a prosthetic. That means the cofactors which are tightly bound and cannot be easily removable. So only after denaturation, if there is a change in the conformation because of the high temperature, high pH, so there is a deformity, then only we can remove this. So these are the prosthetic groups. These compounds are called as prosthetic groups. The metal ion like copper, iron are prosthetic cofactors. So these are called prosthetic cofactors. Some enzyme function only if a covalently bound metal ion is available in its active site. That means there are the enzymes which require the metals at their cofactor binding site, and they are bind covalently. covalently, then only they are active. But coenzymes are organic cofactors that loosely bound. Cofactors are tightly bound and coenzymes are loosely bound. They are organic compounds and some enzymes require metal ions for their function. This is the difference between the cofactor and the coenzyme. Cofactors, it may be metal ions or organic compounds. If they are metal ions, we call them as prosthetic groups, and they are organic compounds, we call them as coenzymes. They are very essential for the activity of the holoenzyme. So here I have listed the metal ions which are required by some of the enzyme. Zinc acts as a cofactor for carbonic anhydrase. Again, it is a cofactor for alcohol dehydrogenase. Iron, it is a cofactor for cytochromes and hemoglobin. Again, it is also for ferredoxin. Copper, it is a cofactor for cytochrome oxidase. So, cytochrome oxidase is present in electron transport chain. Potassium and magnesium, they are the cofactors for pyruvate phosphokinase. So, this is the elaborative description of coenzyme. Is any freely diffusing organic molecule. This is the coenzyme. Freely diffusing organic molecule that serve as a cofactor with enzymes by aiding the function of the enzyme. We call it as coenzymes. Coenzymes is small organic non-protein molecule found in the cell. Coenzymes function as the intermediate carriers of electrons. Intermediate carriers of electrons. So they are very specific atoms or functional groups that are to be transferred during the catalyzing reaction. For example, NAD, nicotinamide adenine, that is NAD transfers electrons in a coupled oxidation and reduction reactions. So that's why it is when it is reduced, it becomes NADH2. Coenzymes are modified during the reaction. Since coenzymes are chemically changed during the reaction, they are considered as second substrate or co-substrate. As I told you, they undergo maybe oxidation or reduction or transfer of protons. Okay, that because of their chemically changing, we call them as co-substrate to the enzyme. On the other hand, since coenzymes are regenerated in the body, their concentration should be maintained inside the body. Most of the B vitamins, B complex vitamins are coenzymes that transfer atoms or groups or atoms between the molecules during the synthesis of carbohydrates. See here, cofactor, and this is the site for coenzyme, and this is the active site or catalytic site. Again, as I told you, it is a coenzyme, and the whole along with this cofactor coenzyme, it is a holoenzyme. These vitamins should be acquired from the diet since they are unable to be synthesized in the body. These coenzymes, most of the coenzymes, they are in the vitamin in nature. Chemically, they are vitamins in nature. That's why they should be acquired through diet, because the human body cannot synthesize vitamins. They have to be supplied through the diet. So since they are not supplied, if they are not supplied, so it leads to other metabolic complications. Okay, these vitamins which act as a coenzyme should be acquired from the diet since they are unable to be synthesized in the body. Again, this is the table which gives the information on different coenzymes and where they are functioning. NAD, nicotine adenine dinucleotide. Okay, it acts as a that is the transfer of electrons, and that is the hydrogen atom in the metabolic reactions. NADP, that is nicotine adenine dinucleotide phosphate, which also act as a transfer for the transfer of electrons or hydrogen atom. FAD, flavin adenine dinucleotide, the vitamin B2, it also for the electron transfer. Coenzyme A, acyl groups. Coenzyme Q for electrons. Thymine for aldehydes. pyridoxine for amino groups, biotin for carbon dioxide and carbamide coenzymes. They act as for the transfer of alkyl groups. This is about coenzymes and cofactors. Next, we'll go through there are some factors which affect the enzymatic reaction. The factors, so we'll study one by one the factors affecting enzyme activity. The first one, concentration of the enzymes. So imagine a test tube where you have provided the substrate and the enzyme. Okay, so if you increase the concentration of the enzyme, so along with the increase of substrate, so you can see the the rate of reaction increases along with the enzyme concentration. If you increase the concentration of the enzyme and also the substrate. Okay, but if the substrate concentration is constant, so it will take a like a hyperbolic curve that I'll explain. But remember, so if you go on increasing the enzyme concentration along with the substrate, so you can see that rate of the reaction also increases. That you have to remember. The rate of the reaction is directly proportional to the concentration of the enzyme. So when pH, temperature and substrate concentration are the constant, but substrate concentration you have to increase. But when pH and temperature, if you maintain constant. So the enzyme concentration, okay, it can have a clinical application in determination of the enzyme concentration and also diagnosis of some inborn errors in the metabolism. These enzyme concentration will be calculated in clinical applications. Okay, so but this is how the rate of the reaction, more enzyme molecule can react with more substrate molecule, so the reaction rate increases. The same thing I have given you. If you go on increasing the enzyme concentration, rate of the reaction also increases along with the substrate more concentration. If you give more substrate molecules, the scenario assumes that there is a large excess of substrate, right? So if you limit the substrate, what happens? The effect of enzyme concentration, as the amount of enzyme increases, the rate of the reaction also increases if there is a more enzyme molecules that are needed. So adding additional enzyme will not increase the reaction rate. Therefore, the increase in enzyme concentration increases, but then it levels off when you limit the substrate concentration. Then the graph appears like this. So if you limit or if the limited amount of substrate, if you go on increasing the enzyme concentration, so there is no sharp increase in the rate of the reaction, because until the enzyme get completely saturated, the reaction rate increases. After that, it becomes a constant. Then what is the second parameter? What is the effect of pH? The rate of the reaction for all enzymes catalyzed reactions depends on pH. So most enzymes exhibit the optimum activity, more activity at a pH level between pH 5 and 9. pH 5 to 9, so almost all the enzymes, they show their optimum pH. So high and low pH value, the optimum value will cause the ionization of the enzyme, which result in denaturation of the enzymes. That we know, some of the enzymes they are acidic, some of them they are alkaline. Okay, when it is in the extreme conditions like in highly acidic and highly alkaline, so they will cause the ionization of the enzymes that lead to the denaturation of the enzyme and the loss of enzyme activity. So effect of pH is that most of the enzymes, okay, so that is between pH 5 and 9, they shows their optimum activity. What is the effect of temperature? So each enzyme has a narrow range of condition it functions. Okay, it works best at the optimum temperature. So most of the enzymes they exhibit an optimum activity between the temperature 30 to you can start from 25 to 40 degree. But there are some enzymes which shows their activity at the extreme temperature also. So as we know, intact polymerase, so that you will study in further classes. Okay, as the temperature drops, the molecule moves slower and the enzymes will unable to function properly. As the temperature goes up, the bonds in the enzyme structure break and the enzyme loses its function. This is called denaturation, that you know. As I am explaining the previous slide, the effect of substrate concentration. So the rate of reaction increases along with the substrate concentration until the increase in the concentration does not affect the reaction rate, and that point we call it as a point of saturation. That means until all the active sites of the enzymes are bound, the rate of reaction increases. After that, if there are no enzymes, substrate molecules will be available, but there are no binding site, so it leads to the point of saturation. The next step is the reaction becomes constant, does not affect the reaction rate. That's we'll study in the Michaelis-Menten equation. Okay. In factors affecting enzyme catalytic activity of the enzyme, concentration of the enzyme and the substrate. It provided that, see here, the enzyme concentration along with the substrate increases the rate of reaction, but with the limited amount of substrate, it becomes a hyperbolic curve. That is, provided that substrate concentration is high and that temperature and pH are kept constant, the rate of reaction is proportional to the enzyme concentration. For any given enzyme concentration, the rate of reaction increases with the increasing substrate concentration up to a point above which any further increase in substrate concentration produces no significant change in the reaction rate. This is because the active site of the enzyme molecules at any given moment are virtually saturated with the substrate, and the enzyme substrate complex has to dissociate before the active site is free to accommodate more substrate. Okay, next it is enzyme inhibitors. Enzyme inhibitors, there are two types, non-specific and specific. Non-specific, which are because of denaturation, high temperature, pH, high acidic, basic alkaline condition, alcohols, heavy metals, all this can cause the denaturation of the enzymes. Okay, but in a specific, maybe reversible and irreversible. In reversible, this non-specific changes leads to the irreversible enzyme, that is enzyme inhibition. Reversible, you have two types, competitive and non-competitive. So that we will study further. Okay. That is, these enzyme inhibitors also are factors which affect the enzyme activity. Okay. Next, activators, the catalytic activity of the enzyme in addition to the substrate concentration is affected by type of concentration of inhibitors, that is the compounds which decrease the rate of catalysis, and activators which has the opposite effect. Activators include metal ions which speed up the reaction, the compounds which are active as prosthetic growths, as I told you, and which provide the stabilization of enzyme conformation of the enzyme substrate complex. So inhibitors are found among the food constants. So again, the factors affecting enzyme activity, what you have to study is starting from enzyme. First one is enzyme effect of enzyme concentration, effect of substrate concentration, effect of pH, effect of temperature, then effect of inhibitors and effect of activators. So metal ion catalysis, they are act as a activators. Nearly 1/3 of known enzymes require metal ions for their catalytic activity, and this group of enzymes include metal ions. Most common transition metal ions are iron, copper, manganese and cobalt. That is, ionic interaction between an enzyme and enzyme bound substrate can help to orient the substrate for reaction to stabilize, where they act as activators. So these are all the factors which are affecting the enzyme activity. And the effect of heavy metals such as lead, copper, mercury and silver are poisonous due to their effects on enzymes. So when a heavy metal ion is present at the active site, a substitution of different metal ion from the original ion can cause the enzyme to malfunction and lose its activity. So this is particularly evident where heavy metals can bind to chelate to, that is, sulfhydryl group in a proteins to form a covalent bond with a sulfur atom and displacing a hydrogen ion. Okay. So this is about the factors affecting enzyme activity. Just go through this video. Just a minute, I'll play a video that show you how to. Just a minute.
Cofactors and coenzymes. Some enzymes need non-protein reaction. These non-protein components are called cofactors. An enzyme along with its cofactor is called holoenzyme. If such enzyme loses its cofactor, then it's called apoenzyme. Apoenzyme is usually inactive and requires the cofactor for its function. For example, DNA polymerase requires magnesium ions for its function. The DNA polymerase along with magnesium ions is called a holoenzyme, while the DNA polymerase without magnesium ions is called apoenzyme. Same is true for RNA polymerase. The RNA polymerase requires sigma factor for its function. The RNA polymerase along with the sigma factor is called holoenzyme, while the RNA polymerase without the sigma factor is called apoenzyme. The cofactor can either be inorganic or organic molecules. The inorganic cofactors are usually metal ions that are required for its activity. For example, the enzyme carbonic anhydrase uses zinc ion as a cofactor at its active site. The organic cofactors can either be tightly bound with an enzyme or released free after the reaction is complete. The tightly bound organic cofactor is called prosthetic group, whereas the organic cofactor that is released after catalysis is called coenzyme. Most of the coenzymes cannot be synthesized by the body and are derived from vitamins. The example of coenzymes includes NADH, NADP, FAD, etc.
Okay, another video I have the factors which deals with factors affecting enzyme activity. Just watch this. The factors which affect the enzyme.
Factors affecting enzyme activity. There are several factors that affect the rate at which enzymes can do their job and the effectiveness by which they can do their job. And the first one is temperature. Enzymes become increasingly effective as temperature rises until a plateau is reached slightly above body temperature. Beyond this point, temperature increase will cause a decrease in enzyme activity as they're denatured. So what we can see here is that at a particular temperature, we'll call it T, the reaction is going on, whatever the reaction is that's going on inside the human body, but it's going on. Factors affecting enzyme activity. There are several factors that affect the rate at which enzymes can do their job and the effectiveness by which they can do their job. And the first one is temperature. Enzymes become increasingly effective as temperature rises until a plateau is reached slightly above body temperature. Beyond this point, temperature increase will cause a decrease in enzyme activity as they're denatured. So what we can see here is that at a particular temperature, we'll call it T, the reaction is going on, whatever the reaction is that's going on inside the human body, but it's going on at a very, very low rate. As temperature increases, the rate of reaction actually increases, and this continues fairly steadily. And at 37 degrees Celsius, we have a pretty good rate of reaction. And this is the rate of reaction at normal operating temperature. Now, if you increase body temperature, the rate of reaction will continue to increase, but not for very many more degrees. And eventually, you'll reach a very short plateau where even if you continue increasing the temperature, the rate of reaction will not increase, and then very abruptly, the rate of reaction drops off to zero. What's happening here is basically death, because at this point, the enzymes that catalyze reactions are beginning to denature, and reactions can no longer take place. So the reaction rate is zero. Now, when you're sick sometimes, your temperature will go above 37 degrees Celsius into this zone, and your rate of reaction will increase, and presumably that's actually going to help. are affected by the pH change, they actually change their shape and the way they interact with each other. And we don't want that happening to the enzymes in our body. So most enzymes function best at a neutral pH where the enzymes won't be denatured. There is an exception, however. Here's where most of our enzymes operate. But we do have an enzyme that operates at about pH 2, and that's so that it can actually function within the stomach. And the name of the enzyme is pepsin, and it begins the initial breakdown of the proteins that we eat. It needs to function in pH 2 because that's the pH of the stomach, and the stomach's pH is actually pH 2 not so that we can use the acid to digest our food, but basically to kill any bacteria which have enzymes of their own that don't function well at pH 2, any bacteria that we may accidentally eat, and we do eat a lot of it. Another factor that influences activity of enzymes is substrate concentration. So when a certain quantity of enzyme is present, increasing the substrate concentration beyond a certain point will not increase the rate of reaction. Here's what happens. When you have a very small amount of substrate and quite a number of enzymes, all of the substrate particles will be in the active sites of the enzymes, and the reaction will take place, and all the substrate particles will be converted to the product. However, if you add more substrate, the enzymes get a lot more busy, and eventually when you have a very high substrate concentration, all the active sites are occupied by substrates, and the enzyme cannot catalyze the reaction any faster because every single enzyme molecule is occupied with the substrate. That limits the speed of the reaction. If you look at it graphically, it looks like something like this. If you have zero substrate, of course you have zero rate of reaction. Doesn't matter how many enzymes you have if you don't have any of these. Okay, but you add a little bit of substrate and you're going to get a pretty slow rate of reaction because you just don't have that many chemicals to react. That would be kind of the equivalent of there. If you add more substrate, you'll have a higher rate of reaction. But there comes a point where adding any more substrate is not going to be helpful because the enzymes are completely busy doing their job. So we call this the point at which the enzymes become saturated. So what could you do if you wanted to increase the rate of reaction and your enzymes were basically occupied with all the substrate they could possibly manage? The only way that you could increase this rate of reaction is if you added more enzymes at this point to accommodate all those substrates. Now there's a great video you should watch. It's the I Love Lucy at the Chocolate Factory video, and it's a great analogy to enzyme saturation. Have a look. Another factor that affects enzyme function is enzyme concentration. Providing there's enough substrate, the more enzyme you add, the more substrate produced, and this limits the overall rate of the reaction. If you look at this graph, there's an interesting question here. Why does it not begin exactly at 0, 0? The answer has to do with the fact that reactions do occur in the absence of enzymes. So if you had zero enzyme and you were doing a reaction between glucose and glucose, you would still get some product, maltose, but it would be very, very little in a very long time period. So the rate of reaction would be very slow. As you add enzyme, provided you have enough substrate, you are going to get a faster and faster rate of reaction. So the reactions occur in the absence of an enzyme, but too slowly to be useful to an organism. Another factor that affects enzyme function is inhibition, and we'll take a look at this one separately in another video.
Okay, these are the two videos which provided additional information on this coenzymes and cofactors and factors affecting enzyme activity. So there is a big problem in playing the video. You can watch this in YouTube also. Okay, keep watching. Thank you for watching. Thank you.